Hello, my name is Jeremy Dasen. I'm an assistant professor at the NYU School of Medicine. A fundamental question in the neurosciences is how the remarkable diversity of cell types are generated during development. This study addresses the question of how motor neurons become regionally specialized within the spinal cord. These findings may have important implications for understanding the evolution and function of the vertebrate motor system. These images show the patterns of axonal projections in mice where motor neurons are labeled with GFP. Limb innervation is characterized by a complex pattern of projections, with each branch targeting a unique set of muscles. In contrast, the body wall has a much simpler pattern of innervation, which is reiterated over multiple segments. These patterns of innervation are reflected in the organization of motor neurons within the spinal cord. There are two basic levels of organization. The first is the segregation of motor neurons into columns, which occupy distinct rostrocaudal positions. Limb levels contain the lateral motor column, or LMC, which is required for locomotor functions such as walking. The intervening thoracic levels contain the hypaxial motor column, which is involved in respiration, and the preganglionic motor column involved in autonomic functions. The most diverse motor neuron populations reside within the LMC, which can be further divided into over 50 distinct motor pools, each pool targeting a specific muscle in the limb. Hox transcription factors have central roles in the specification of motor neuron subtypes. At forelimb levels, HOX6 genes specify brachial LMC neurons, HOX9 genes define thoracic fates, and HOX10 genes specify lumbar LMC identity. Within LMC neurons, the activities of nearly 24 HOX genes control motor pool fates. One question this study addresses is how so many HOX genes become restricted to limb levels of the spinal cord. HOX expression in motor neurons is thought to be defined by cross-repressive interactions between HOX proteins and HOX genes. This image shows a top-down view of the spinal cord showing mutually exclusive expression of HOXC6 and HOXC9.
To begin to address these questions, we analyzed HOX mutant mice. This revealed that a single thoracic HOX gene, HOXC9, acts as a global determinant of motor neuron organization. In HOXC9 mutant, expression of genes that define thoracic motor columns are lost. For example, expression of NOS is lost from PGC neurons, and ER81 is lost from HMC neurons. Instead, thoracic motor neurons now express markers for brachial LMC neurons, including HOXC6 and FOXP1. Thus, in the absence of HOXC9, the entire thoracic spinal cord is converted to the identity of the limb innervating populations. To assess the consequences of the transformation of thoracic motor neurons to a limb-level fate, we bred HOXC9 mutants to line in which all motor axons are labeled with GFP. We find that projections to the sympathetic chain are lost, with most of the ectopic LMC motor neurons targeting intercostal muscles.
So based on these results, we decided to address the mechanism by which HOXC9 repressed HOXC6 expression. So we used chromatin immunoprecipitation assays to determine whether HOXC9 binds directly to HOXC6 promoter sequences. In this assay, spinal cords from brachial and thoracic levels are dissected and fixed in formaldehyde. The DNA is then fragmented by sonication, and regions bound by HOXC9 protein are purified using a specific antibody. So using PCR, we found that HOXC9 binds to HOXC6 regulatory sequences at thoracic levels. To further define HOXC9 binding sites, we also performed ChIP-seq experiments on ES cells that ectopically express HOXC9 and have been programmed to motor neuron identity. This analysis revealed that HOXC9 binds to multiple HOX genes within the HOXA and HOXC clusters, including each of the 8 HOX genes expressed at brachial levels. This work was done in collaboration with laboratories at Columbia University and MIT.
This group of HOX genes are thought to be required for the specification of motor pools. Many of these pools can be defined by transcription factor expressions such as PA3 and SCIP. In HOXC9 mutants, all brachial HOX genes are de-repressed at thoracic levels, and there's an overall expansion of these motor pools. Our data indicate that HOXC9 has a central role in organizing the motor system. HOXC9 appears to be essential in restricting the diversity of motor neuron subtypes to limb levels through directly binding and repressing a large cohort of brachial HOX genes. The absence of HOXC9 at forelimb levels allows motor neurons to express multiple HOX genes and in turn generate a diverse array of motor pool subtypes. mutation of HOXC9 causes a derepression of HOX genes in other cell types, including ventral interneurons and mesoderm, HOXC9 may therefore have a more general role in specifying the regionally restricted subtypes essential for the emergence of motor behaviors through global regulation of HOX expression patterns. Taken together, our studies reveal a novel mechanism through which the regional diversification of motor neuron cell types are established through the actions of a single HOX gene. These findings may be relevant in understanding how other cell types of the nervous system are specified through networks of transcriptional repressors.